So continuing with our nervous system discussion, we're going to look at the way that we integrate all the circuits of our brain and nervous system and how we use the synapse to communicate between the pre-synaptic axon and the postsynaptic structure, which could be another axon, another neuron sending a message ahead, or it could be an effector. And if you recall, our effectors are smooth muscle, cardiac muscle, glands, and of course skeletal muscle, which we just finished. So the difference between myelinating or not myelinating is really a matter of distance and speed. If we want to just make a short communication, these tend to be our interneurons. We're just going to whisper between two neurons, packed closely together in the spinal cord or the brain. We're going to send a message to the first one. There'll be an interneuron to collect those. Remember, we have the excitatory or inhibitory potential, so they don't say, hey, I want to send this message on, or I don't. And then it'll pass it ahead. Those tend to be these continuous conductions. They're unmyelinated, and we just roll that exchange of potassium and sodium from the one end to the other. Remember, it always goes from the axon hillock, or the base, to the terminal end bulb. But if we wrap it in myelin, there's two types of myelin wrapping. In the central nervous system, it's done by the oligodendrocytes. So it would look like this, but they'd have body parts between them and no cell set up in the myelin itself. So the nucleus would be down here, and they'd be reaching out and grabbing. That is how you can tell this is a peripheral nervous system set up with our Schwann cells, or also known as neural lemmocytes, because they're wrapped individually and they each have their own nucleus, so they're each an individual little cell. So this peripheral axon, we're going to notice that we call it saltatory conduction. We only interact with the extracellular fluid at the node. And the sodium-potassium exchange happens here, so we flip the current. Remember, we begin with a current that is more positive outside than in. And the negative inside is specifically negative 70 millivolts. So as we gain excitement and we're ready to send a signal, when we hit a threshold, which is negative 55 millivolts, we flip the entire structure, sodium rushes in, potassium's kicked out, and we become positive 30. That will send the signal down, but unlike here where it's just going to run, here we're going to hop. So we hop between the nodes, and that's known as saltatory conduction. That speeds us up significantly because we don't have to go that full length, we just jump. So these are for the motor neurons that are out in the body, and any neuron that has the distance to travel, we're going to make sure that it has a sheathed axon. And again, the white matter that we'll encounter in lab, and we'll talk about in here, is myelin. So whenever you see a white matter portion of the brain, or spinal cord, or the nerves, always myelin. So it's a huge part of how we make things work. With the continuous, as I mentioned, we're just going to run along the, I always go left to right when I position it, because that's how we read. But it obviously go in any direction, up, down, left, right, back and forth, but it only goes one way. So it's always beginning signal to the end of the signal. And the depolarization, repolarization happens along the way. Just remember these terms to be clear on what's what. Depolarizing is when we take it at its beginning, we push it up to the threshold, and then it depolarizes all the way. Repolarizing resets it to rest, and then hyperpolarization drops it below the line. So it takes longer to get it back up. So we don't worry about hyper here, because that's just demonstrating what happens in a quick shot from left to right in this case. So repolarizing is when we're going to switch. And depolarizing is when we make it send the actual potential, and we go back to the way we were. That's repolarized. So what they're showing you here is that every time I depolarize and I'm moving along, I'm already repolarizing behind it to clean up what I just did and set it up for another. The chain reaction begins on the one side and always travels to the other. And these channels are what we call voltage gated. Voltage gated are going to pull in these sodium ions that are positive, and that's going to begin at the hillock, which is here in this case, and go toward the terminal end bulb, which is the other way. Because of these repolarization happens behind it, we can only send the signal one way because we've already reset back behind, and it won't be able to go backwards. That keeps us moving in the direction we need to. And the refractory period that we mentioned earlier, which is that brief period of hyperpolarization after we send the signal, is again helping us reset, so it keeps us moving in one direction. In saltatory, however, we are going to concentrate the channels in just these areas nodes. and these little nodes are going to have chock full of the way to pull in the sodium and release the potassium, so these voltage gated channels are ready to go. And the moment that we hear from over here, we're going to jump, 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 and that speeds it along at a much quicker rate. And again, the sheer amount of channels make the voltage change so much quicker. So the speed of this oscillatory conduction, or the myelinated neuron, is key. This sends it to the next node. The action potential, that's for AP, in case you need a reminder on that, repeats it from node to node, and it is much more efficient. We don't have to use the ATP so much to maintain the rest of the potential, because we're only messing with it at those nodes. And this is how we're designed to be as efficient as possible. And this is a big part of early childhood. We're developing the brain networks, and we're myelinating the brain, and that's the major growth that occurs from in utero till our first couple decades of life and growing all that. So when we come up to this visitation between neuron and neck structure, the ending of the signal will always be at an axon terminal, and the axon terminal is going to hold the vesicles with the neurotransmitter that we've made somewhere up the line, and we're bringing them down here. And the neurotransmitters that are in here, we've talked about acetylcholine so far. There's others, of course, but we'll just draw some dots in here representing the neurotransmitters that are going to be released. The space between this and the neck structure is known as the synaptic cleft. And the cleft is a microscopic, beyond microscopic, minuscule space that allows the chemicals to pass, but these don't actually touch. The second structure, which up here we call it presynaptic, and when we get into, I don't think you hit this till AP2, but we'll talk about how we're going to move signals through the autonomic system. An autonomic system will always have a ganglion in the middle, which is going to send something to a ganglion and then to another neuron. So we'll call those pre and post ganglionic. But it's the same idea. Pre and post synaptic is before I get there, and then who's after. So the postsynaptic structure can be, as I mentioned, another neuron or one of the effectors, which are smooth, cardiac, skeletal muscles, or a gland. This must have receptors on it for that particular neurotransmitter. And this presynaptic neuron can make different neurotransmitters. He doesn't have to just have just one. So I can have different receptors for different ones here, but I can only release one at a time. So whatever the message is going to be, that's the neurotransmitter that gets released. And as we talked about way at the beginning of the course, we bring a vesicle to the membrane, they fuse, we use ATP to dump, and we release the chemicals into the synapse. They land on the appropriate receptors. Receptor signals a change in the membrane, but these neurochemicals do not enter, and then they're often recycled or changed another way so we can reuse them again. We've got to get them out of the synapse though, because if they stay in the synapse, they will continue to stimulate this structure. So it's a quick hit and then we pull them out. So the presynaptic neuron will release the neurotransmitters, and those will bind to the postsynaptic neuron dendrites or body. So if this is another neuron, where all these receptors are located are either on the body itself or on the numerous dendrites that are coming off, and that's where we're talking. The single axon going out will always send that way, so we're not interacting with that here. So it's similar to the initial thing that I drew where we have an incoming sensory and outgoing motor. The incoming sensory is this part of the cell. The brain of the cell is our central nervous system, or integration center. And then we go out the motor side, which in this case is that. Remember that the excitatory or inhibitory, which is the EPSP or the IPSP, are going to change how the voltage is going to react. So if I send a lot of inhibitory, I'm going to bring it into a hyperpolarization where we're less likely to do anything about it. So we're saying, don't worry, nothing to see here. But if I send excitatory ones, I'm going to raise that and start to depolarize it and say, this is probably worth my attention. And if I hit that magic threshold of minus 55, even if I just tickle it or if I push way beyond it, I get the full blast. There's no small axon sending action potential, it's always the same. So, and then just, you don't need to know any of these numbers, but it just gives you an idea of how complicated the system is. And if it's just a typical spinal motor neuron, we can have 8,000 different synapses on here, from 8,000 different cells talking to it from the nervous system. But the cerebellum, which we need tiny cerebrum, the back of our brain, where we process all the intended movements we ever have and critique ourselves. The little cerebellum is our voice in the back of our head, literally, that says, "No, you didn't do that right, try it again, draw it better. Don't be so clumsy." There could be 100,000 connections talking to a single neuron to say, "Hey, I need you to really pay attention to this incoming information." Another thing we'll see in a moment is the role of calcium. Just like we saw calcium playing an important role in allowing the muscle to interact at the protein-based level with our myofilaments, we're going to see calcium is going to be entering here, and calcium is what actually causes these synaptic vessels to dump. So not only is calcium, if this were a muscle, key in our cisterns to release just in time to make our actin and myosin interact, it's floating up here in our extracellular fluid, and it's drawn in through the calcium channels, and that is what actually makes the synaptic vessels dump. And there it is. I thought that was coming up. So when the action potential arrives, you see the little arrows here coming down. When I hit this end bulb, that will open the voltage gated calcium channels which are around here. Remember the 3 gated we had? It can be modality gated, where we're going to receive information from the outside environment usually, or inside, but it's a sensory modality. So it could be light, sound, chemical, smell, touch, taste, pressure, any of that. The voltage ones are based on opening at a specific voltage change here, and we're going to let in calcium at that point. Calcium will diffuse in, they will bind on the synaptic vessels as you see them wrapping around it. And they're consistently drawing those little calciums as little pearls, which is kind of neat. They're white crystals of rock-like material, so here they hit. And that will, again, if you imagine being heavy and pulling things down, that's one way to look at it. We mentioned that on the troponin, they kind of tip it and open up the spot. Here they're kind of making these things drop and pop them open. It's like glitter coming out at a party or a confetti. So down they go. Exocytosis releases neurotransmitters to the synaptic cleft. All these steps are not that different than what we talked about before. We're just zooming in on that one step where we just said we release the neurotransmitters. Now we're just telling you the specific parts. Whenever you learn these, and I know going through them for the last test is challenging, all those weird words and these orders and things, I teach it as a way to prepare your brain for your future studies when you're going to have to learn intense protocols and procedures for sterile fields and inserting needles and catheters and making sure everything's done just right, and you'll be tested on that in a real scenario. If your brain can work through step by step, that helps prepare you and build that knowledge. So just working at this, the same way we did for the muscles, just saying this cannot happen until this happens, and you work through it, practice it, say it out loud. It's no different than you telling me your favorite recipe and me trying to do it without listening and making a mess of things. You say, no, you have to do this before you do that. So just learn this, and as we did with muscle, and if you're still struggling with muscle, you get one more shot at it at the final TA. But you get a chance to just walk through it. Say, well, I cannot do this step until I've done that step. And this is the one that started it. And the initial thing was the summation of the excitatory potential on the other end of the neuron, and just keep saying it and walk it through it until it makes sense to you, the way you can put it out. And then again, the rules that we took back from the very beginning of the semester say that the reason why we have proteins embedded in recognize our old friend, that phospholipid bilayer here, the reason why we have proteins embedded in here is because they're waiting to receive signals from the outside world to say, hey, it's time for you to do your thing. Whatever my thing is down here is probably related to protein. I'm either making protein that I'm going to submit down the line, I'm using my protein fibers, filaments to pull, or I'm making a protein that I'm going to submit into the blood and secrete it as a hormone. So it's basically stepping up the work of whatever's on the other side. When they arrive, they will trigger a graded potential down here. And then whatever happens down here depends on if it's another neuron. This could be an excitatory or inhibitory message saying, let's go or let's not. This could be a muscle that, like we said, is going to do the same steps we just went through. Or it could be a gland that will secrete or stop secreting its substance. Because we can control the autonomic system with a fine-tooth comb saying, yes, do it or don't do it. Whereas the muscle, as soon as this hit, I gotta contract, I have no choice. But if I'm using a gland or another neuron, I can say, let's send you some excitatory signals to make you do your work, or inhibitory signals to make you stop doing your work. And that's kind of a neat way to control. And that's where we get into the whole parasympathetic versus sympathetic. How are we talking to our body parts to say speed up, slow down, and so on. So these neurotransmitters, once they are released, they're gonna land here, they don't enter the cell. We're gonna do about 3 different things. We're gonna break them down, recycle their parts. We can diffuse away, float off into the world, or we can recycle them. We immediately reuptake them and use them again and again. So the reuptake, we'll talk about that in a minute and how medicines work, but most of our modern pharmacology is based on this synapse and saying I can substitute something that will stimulate this the same way as if the neurotransmitter is there. I can block them from hitting here in the first place, or I can leave them in the gap and make this feeling continue. So there are many neurotransmitters. For this course, you don't have to know them, but just an example of how they're organized and how they, some of their names and their formulas, if you're curious. You've heard of acetylcholine already. You maybe have heard of epinephrine and norepinephrine, but they're a huge player in our system of sympathetic nervous stimulation. They are adrenaline. So the other name for epinephrine is adrenaline and noradrenaline. Dopamine is our do it again chemical that hits our brain when something exciting happens that our brain is curious about or wants to see it again. So whenever you hear dopamine, the message is do it again. Whatever we just did, do it again. So our phones work on us, and the slot machine, and all the things that we just can't stop doing. And then serotonins will bring us a peaceful, easy feeling, to quote the ego. And histamine is used to work on an allergic reaction. Some of us may be hitting that histamine time of the year as the pollen comes out and our body starts acting a certain way. So these are examples of biogenic amines that come out in your synapses. And then GABA, we can replace its effects with alcohol. So alcohol binds to the GABA and gives us a depressed feeling. So that is a problem with long-term alcoholism. And then glycine and aspartic acid are other examples of amino acid-based neurotransmitters. So again, just info, but there's many of them, and that's the role of us. They're all made of the same chemicals that we always started with: carbons, hydrogens, oxygen, nitrogens. It's just how we combine them. So therefore, you can see in the labs and in the pharmacy, biology labs, they're creating these things that are very similar by pulling from animal sources and recreating them in our version, or just building them outright as a medicine, what we call it. So there's over 100 different types of neurotransmitters hanging around out there. As I mentioned, a single neuron can make multiple types, but it will only release one at a time. And then they will hang out here in just a fraction of a second, make the signal continue, and then break down, but they can hang around longer. And that's again where the medicine and drug changes will affect how our neurons talk to each other. So here's all the examples. Again, I don't need you to know this stuff, but I think it's pretty cool to see how we've calculated and figured this all out. Remember that an amino acid is our base of a protein. All we do is modify them just a little and turn them into, and here's their original forms, the amino acid form, and this is what we turn them into to make parts of our body. So when you say, and this is where it got its bad reputation, tryptophan's in a lot of things, but the idea that it was a protein that we might get in a meal that we had at Thanksgiving, for example, and it might turn into serotonin, which makes us feel, so it's not actually that because all meats have it, but it was that idea that, well, that's why we're tired. Well, it's probably because you ate too much, but that's all right. The amino acid ones, as we mentioned, they stay as the original form. And then the neuropeptides, which aren't here, a ton of them, which are rich strings of amino acids, and those are also include our endorphins. Endorphin is a rush of feeling. It reduces pain, it gives us euphoria. So those are particularly dangerous when they're in pill form, like our, as we know, the OxyContin and all those things are based in that. So when we get a neurotransmitter hanging out in here, we have to stop this message before too long, or else this structure will get fatigued. So while it's there, we can't send any more messages, and we have to take it away from the receptor. So how do we get it off of these receptors once they land? We generally will break them down by an enzyme. So we already mentioned acetylcholine as the main communicator between neuron to neuron, but here neuron to a skeletal muscle. Acetylcholinesterase will break that down and recycle it to go back into its parts and rebuild it. We can reabsorb it directly as it is, and the monoamine oxidase, MAO, will pull them right back. You may have heard of an MAO inhibitor that we use to help with heart conditions, and that will keep that from going backwards. And then they can also diffuse away, but the astrocyte, who's that star-shaped neuroglia over here, will take it back and put it back in again. So we're not wasting anything if we can help it, but we're reusing it in multiple different ways. The transmission that happens here can be modulated by what we call a neuromodulator, which will change how many receptors we have, which is saying the phrase we use in neurology is the neurons that fire together wire together. And what we mean by that is over time, we can change how many receptors we have, how much dumping we do, and how many axon terminals we have. We can't change the number of actual neurons, but we can make the terminal branch more, the more we need it, and build a stronger connection at the receptor level. We also have different components in here, like nitric oxide, which will help make our neurotransmitter release better. So this is kind of tweaking brain chemistry and neurochemistry to figure out what's our optimal performance. Then here's the idea of what we refer to as the medicines or drugs, both good and bad, and their main purpose is to slide in here, trick the structure here that it is a neurotransmitter, and therefore we get an effect. So the MAO inhibitors we mentioned, they will keep the amino acids and monoamines in there, and that will help over the long term. SSRIs are a very common anti-anxiety, antidepressant brand of medicine, or type of medicine. And if we look at serotonin as our happy chemical, I gotta spell it right, hold on. There we go. And serotonin is going to show up in a field and say, hey, I need this neuron, put him over here, he's feeling a little sad. I'm going to put some neurotransmitters in here called serotonin, and he's going to feel a little happier. But I'm going to take him back right away, and I'm sad again. So if I can reuptake, inhibit, stop it from going back, and it'll stay in the synapse longer, and I'll get a little bit happier. So we work that balance of what's the right dosage for you when you go through those kind of things, and it changes our ability to interact at that level. So this shows a normal pathway. Serotonin comes out, the pre, it lands in the post, and then it's put back. And it stops. So if serotonin is making you feel good, and you stop feeling good, or you're not making enough of it for whatever reasons, we can use this drug to say, hey, instead of going back, let's block you from leaving, and now we're going to stay there. So we stop that last step, the reuptake step, which is why we call it reuptake inhibitor. Just remember, inhibit always means to stop doing what you're doing. And these are all the brand names, but that's a example of SSRI medications. Again, nothing you need to be tested on. I'm not going to, it's not a pharmacology course, I won't ask you which ones are, or what amino acids are involved, but just so you know how this works. And again, numbers, don't need you to know that, but this is what I meant by synapses. The number of synapses I can make within a given part of the brain is astronomical, to quote the astrocyte. Cerebral cortex can have up to 40,000 synapses between cells, and the estimate is that in a typical brain we have 100 trillion different ways that our neurons can communicate, so it's internet level. And this is really how we use these at this level, is how we explain memory and understanding and thinking and all these things that we use. All it is signals. So we're basically taking sensory information, translating it into a chemical signal, and then summing those together and making meaning out of them. And the meaning part is really what makes the brain learn and grow. And we can explain this the best we can at this level, but it's still a frontier of understanding of how we actually do all this in this little space up here. But we can break it down first by saying some stimuli are considered qualitative, and that's something that we put a quality to. Feels good, feels bad, feels salty, feels sweet. It's which ones are fired and what they're using. So if I'm using specific ones, let's take the tongue, I'm going to draw a tongue. But if we go in the tongue, we have different receptors that are looking for different types of flavors, which are just chemicals, and they're called taste buds. So certain taste buds that are distributed might get triggered if you eat something salty. So if those fire, they'll go to a part of the brain that identifies salt. And you'll say, hey, I think I just ate something salty. But if they hit a different one, they'll send a sweet signal or a umami signal. So that's which neurons are active. But if we say quantitative, we're going to hit a ton of them. So if you eat something that's really salty, you're going to send more signals from those same neurons, and that'll say, wow, quantitatively, I've got a lot of salt in my mouth right now, versus I just tasted a little piece. And our ability to detect sensation is, we can detect the slightest flavor difference, set on the right spot. The other thing that will happen is we're going to send the signal more often. So this is where pain comes in. Pain is a warning sign that something's off, and it's really hard to ignore that feeling. So the rapid firing rate from the cell down here in distress will continue to bother you. Like, that really hurts. And it'll keep sending the signal. And then we go back to the step, well, what if we take a euphoric medicine and blocks this? Well, we don't feel it anymore. It's still hurting down there. The cell's still in distress, but we blocked it. We feel better. But then when that wears off, we don't feel so good. So that's the whole idea of that. The other piece that we can kind of break down and describe, and this is really hard to visualize, but it's describing circuits like you would describe it within your house for lining up. Like I've got when I moved the desk, I noticed there's a multi-prong setup there where I can plug in a bunch of different things, but it's all coming out of one plug in the wall, so that's kind of how we're looking at these options. So do we converge on a single neuron? Do we send a bunch of neurons to talk to a single one at the other end? Do we go back and forth, or do we do something totally different? So this is what we call a neuronal pool or circuit, and these are just ways to explain how we understand how things are happening up there. If we're converging means to come together. So we saw this in the ESPS and the ISPS. A single neuron at his dendrite level receives many inputs from other sources, and he sends out a single output. The single output is waiting until he hits threshold. So we use this to say, I'm going to talk to from a bunch of different sources into a single output. In this case, in the example, let's say the output is the salivary glands, which are exocrine glands in the mouth. They will respond to several different things. So the example of the input would be, oh, I see somebody's cooking. And I smell somebody's cooking. And I taste somebody's cooking. The next thing you know, we're salivating. So we get multiple inputs to receive the signal that is going to give us the same output. And then if we go with Pavlov's, I hear a bell and I salivate. So that's huge in understanding how the brain works when we realize it's not actually, doesn't actually have to be, if you go into psychology, the actual signs and sights and smells of cooking. It's something that you associate with it. So you may go, we associate time with meal too, we get hungry, but we're like, it's time to eat. That's what we mean by converging. If we're diverging, we start with a single idea or thought, and we have a network of things that happen. So this is how we run our body. We don't have to think about anything we're doing at any given moment. We wander around, we drive cars, we do all kinds of crazy things based on input of, I need to get this job done, and all these outputs are saying, these are the motor networks that I've set up over my lifetime to make this action happen. So every word you speak, every step you take, every breath, no, I'm gonna go there. But you are ready to do everything, it's all pre-programmed. When it goes wrong, when you have a brain injury, stroke, or some other connection right between here, then we have to relearn and realize the danger of that. But it's redundant. We have a lot of redundancies built in. We have two different hemispheres that helping. In the end, it's the best system that we know of, but it's a fragile system because these neurons can be killed with deprivation of oxygen and glucose, and that's where we lead into the rehabilitation process. If you lose part of the brain for damage, we can work around it, find some other guys that are going to talk to this network, but it depends how much damage there is. But this is how it works in diverging. This is, again, how we control our body. The reverberating circuit is how we imagine memories work. We're going to send signals back and forth and get a little loop going, or reverberating. And that little loop or reverberation will help us maintain either a constant behavior, such as sleeping. We don't need to be awake to breathe, so once we're sleeping, the circuits just run and say, just keep breathing. And this is in between the pons and the medulla. We'll do that next year. But at the base of the brain, there's a little pons, we'll run them very soon. We've got a pons and a medulla here. They're going to have a reverberating circuit that talk to the diaphragm at the base of the lungs and say, just keep breathing. And that is an example of that. They have redundancy, so saying if you forget or if something goes wrong, I still got you to remind you. This also helps us hold thoughts over time. So this slips as we go, like, why did I go in that room? Or what was I going to say? It's going to tip my tongue. That was frustrating. But that's what we're doing here. We're saying, I need to remember this. I'm going to hold on to it. and it allows us to experience what we call the flow of time. We know that we're present in reality at this moment because our brain is running a circuit saying, okay, everything's checked off, we're here. And it's kind of crazy how if you go that deep, you can say everything that we experience is just these guys chattering inside a dark cave here. And we assume it's reality, but anyway, let's not go there in this course, but something to think about. Philosophers have debated that for eons. What is it? What are we doing here? What is it? What's real? What's not? And then when virtual reality shows up, we can get lost in another world that we can create for ourselves and what we do in our own imagination, in our own brains. And then finally, we have this idea of some of the higher order thinking that we possess as humans. And this is generally going to be frontal lobe stuff. So when we break the brain into parts, whenever we talk about higher order or advanced or the thought processes that's generally happening in our frontal lobes, which are most developed in the human and the dolphin, but we'll leave him out of the story. So we're going to have inputs going different ways to the same output. So you'll see different ways they're going to get there, connecting over here, but they're all going to end up at the same output, but these guys are going to talk to each other along the way. So that will give us the same signals coming in at different times and help build a idea of consequence, action and reaction. We think through different things when we have a developed frontal lobe to say, well, what happens if I do this? And if I don't do this, what will happen? And we run through all those scenarios in this kind of setup to arrive at a decision. So that's what we think those are happening. And that's our story. So on that slide, show the majority of stuff I thought you did on the test have to do with the conduction and the neuron activity at the synapse. Because each of these topics that we're hitting is an entire course in itself, or a doctoral degree in itself. This is the deep stuff. And we're just going to give you enough of it to pique your interest and then move on to other subjects as we move through it. But the brain and everything that happens here is the huge frontier yet of our research and our ability to understand what is humanity. So most of what taking the brain apart, looking at the parts will be lab, but we'll give you the regions here that will help underline what you're going to do in lab and try to make sense of all the different parts. You'll learn your 12 cranial nerves in lab. You'll set up images of maybe the actual brain that is going to be dissected or the models, and you'll look at it and try to identify things. So let's start with the first outside look at it. We pop it out of the skull, we peel off the outer skin, which is called the dura, and we see something like this. Yours will look, when you do if you do one already in human bio, the sheep brain will be a little flatter, cerebellum will be back here, and the brain cells going that way because they go around the world this way instead of us, where we're going down. You'll see a much smaller cerebrum compared to the cerebellum, and you'll see much larger olfactory bulbs, which we'll name later. They're huge up here, which are controlling their sense of smell, which they use to identify the world. We use ours, but nowhere near what the animal world does. But the interesting thing is that whatever creature we take apart that has a brain, we can find parts that look familiar. What we call the reptilian brain, which is deep inside here, even the humble little lizard snakes crawling around, they have all that part. So that's why we call it that. But imagine that as different species evolved in the animal kingdom, we developed different parts of the brain where we can identify similar parts on almost anything that we crack open the skull on. So we'll start with the four major parts as we divide them. You can only see three of them in this picture. So we come up from the spinal cord and down to this body through this brain stem, which a minute ago, just before this line, we called the spinal cord. Where it separates from in naming and in function is at that foramen magnum at the base of your skull and the occipital lobe. So occipital lobe here, coming up, spinal cord, oh, now I'm brain stem. When I get out, you're going to notice all these little yellow guys, which are the nerve roots for what we'll call the cranial nerves. And many of them are here, there's more up here and in there. We'll name those in lab as we go. The next thing you'll see is the back of the brain hangs off in the cerebellum. And that means a little cerebrum. So you'll see it's the same kind of setup here with these folds and gyri, but we don't call them gyri here, we call them fold, like a book page, it's like a folio. Here we have these lumps and dips. The lumps are called gyri and the dips are called sulci or sulcus. This whole thing is known as a cerebrum, and it is the majority of our brain volume by far. But half of the neurons we have exist back here. So again, this is the little brain, or the little cerebrum, but this is the part where we said they have thousands and hundreds of thousands of connections, because this is where we refine everything we do. So when you work with someone with cerebellar issues, they lead to what we call ataxia, which is coordination problems. They can't move their body in ways that make sense. And if you just notice by the way I was stumbling, this is where we get hit with alcohol. So when none of you would have done this, but if you're ever pulled over on a roadside sobriety test, the things we ask you to do, I'm never giving a sobriety test, but in lab we can do cerebellar practices, close your eyes, touch your nose, do all the walking. This will tell us, is there cerebellar dysfunction? Regardless of whether it's your ataxia from drinking alcohol or other sources, you shouldn't be driving a car if this isn't working well. The other thing, of course, alcohol affects is your frontal lobe inhibition, saying, that sounds like a good idea. Of course I can drive. Anyway, deep inside, we have a piece called the diencephalon. Remember, cephalon means, cephalo means the brain itself, so diencephalon is inside. So I usually visualize it like this. We're coming up that foramen magnum. We come up and we're gonna blossom into two little balls like that, and then we wrap everything else around it. Those two little guys are in there are called a thalami, or thalamus, and then in there is the parts of the hypothalamus that are going to dangle down. What we're not seeing here is a little pituitary gland that would sit right there. He's going to be gone by the time you get your brain in lab, because we can't preserve that very well. Once you cut the brain out, we lose it. He sits in the sella turcica. Remember him from the sphenoid? So there's sphenoids here, there's a sella turcica, and the pituitary gland lives right there. So when you take a brain out, we often lose him in that. His little infundibulum or stalk is snapped off. So we don't generally see him on we see him on models, but not on a dissecting brain. And there's my man chilling with his brain in his head. So he's thinking cool thoughts. What this blue stuff represents is the fluid our brain floats in. So we're floating up here, sloshing around, and that's for cushioning and transmission of information, and it keeps it clean and flowing, and it's called the cerebral spinal fluid. The cerebral spinal fluid provides that medium in which it all exists, and it circulates all the way down to the base of our spinal cord, back up and around. In the center of the brain, there's 4 chambers we call ventricles, which are big areas where it is formed. We take it out of the blood. All fluid is going to lead us back to the blood eventually, because the blood is our major highways. So we sort through that, pull out just the water part, and make a sterile fluid called cerebral spinal fluid. But we gain it through the blood, and that's the ependymal cells we mentioned before in the cerebral family of microglia, or neuroglia, I should say. Microglia is a specific type. And they're little cilia hairs that wash this around. So if we work our way from outside to in, we have a solid skull. Underneath that, we store a little fat and some veins, and that's called the epidural space. Then beneath that, we have a dura. And the dura is a tough coating that surrounds brain and spinal cord. So that looks like this. We wrap it around the brain, we dip down the center, because there's no brain matter there, and we come down through our spinal cord, and we form a little sac at the bottom of the spinal cord where all this fluid is going to float. So this area, and all the fluids floating around in here, has brain and spinal cord through it. So from outside to in, we're going to go skull first. We're down here, here's our foramen magnum. Down here, we're going to have a vertebral column, so these are protecting it here. So bone first, we should say. And beneath that, we have what we call an epidural space. So it's got a little fat, a little veins. Epidural, you may remember, if you've heard of it before, is when we're going to put a needle in just before the thecal sac ends, but after the spinal cord ends. So you'll see this in lab, but this is our brain. I'm going to go green here. This is our brain up here, and our spinal cord down here. The spinal cord will show up short of the full thecal sac. He stops around L1 and L2. So the spinal tap, or the epidural, is going to go beneath that. I went too deep in my epidural. I just failed nursing. There we go. Let's go back. So there we are. We're going to hit the epidural. If I pierce that, oh boy, I've caused some trouble. So I go in my epidural space, and then I can secrete medicine in here that will numb, and they know exactly how high to go, how hard you're going to numb. So if anybody's ever had one through childbirth or had a spinal tap drawn because you're worried about meningitis, which is mentioned here, that's where we go in. We don't want to pierce any of this material. After that, we hit the dura, which is a tough coating. So dura sits, well, I kind of drew it already right there. That's the sac. Beneath that, we have a subdural space. If you've heard of subdural hematoma, I hit my head hard enough that I'm bleeding here. Now I've got a real problem because this blood is going to push there, and I can't go out here because I'm solid bone. So subdural hematoma is a crisis where we're going to have brain damage. Then we hit the arachnoid layer. The arachnoid layer is where all this fluid floats. I'm just going to give you this little acronym. From in to out, we pad the brain, dura, arachnoid, and pia. So the brain is padded from inside to out with this acronym. Pia means gentle. The last word, mater, means mother. So pia is your gentle mom. She cradles the brain. When you work on the brain in lab, you'll notice it's like a saran wrap, little tiny, tiny skin. And you hardly even notice it, but it's this little peel that comes off. And you're getting into the tissue. The arachnoid, you're really not going to be able to see much of that because it is spider-like strands, and it's not going to preserve well when you open the dura. And all the fluid that's in the preserved brain would be the formaldehyde solution, but in our living brain, it's the cerebrospinal fluid that's floating. And that's where the CSF is floating in the arachnoid region in here. And then the outer tough coat, which you'll have to cut through with scissors or a knife. Peel that open, it's like a little leathery coating. That means tough mother, like durable. So the dura mater, you can call the arachnoid mater, but it's often just called the arachnoid, which means spider-like. So you got little spider webs. So it goes mom, spider, tough. In that order. We made a blood-brain barrier back when we talked astrocytes. So anything that gets in here, we try to protect it from getting in there by the astrocytes saying, you're not allowed to touch my neurons, because if you get in my neuron, he dies, the whole system's falling apart. So we have multiple ways to protect this brain. But as I mentioned, the downside of having a solid skull, which we need, is that if damage happens inside, we can't get that to stop. We have to go in here and open that up and evacuate that. So you'll see burr holes in brains. You'll see people removing pieces of tissue to get out a severe brain injury and have a gap in the skull. So the craniotomy or craniectomy will remove pieces, and then we can break that pressure. When you're an infant, you don't have a solid skull. It's tissue. So babies that are born with brain fluid, we call it hydrocephaly, their head will swell. And that's usually related to spina bifida. I'm getting way off top, just throw it out here while we're here. Spina bifida says my dura is going to pop through the spinal column because I didn't finish building my spinal column because mom was lacking folic acid at a key time. And then I've got this fluid set up here, and then I'm going to blow this whole thing up. So those are some things you might see if you're in maternity nursing. We're going to want to worry about, why, yikes, we got to deal with this right away because this poor little guy is in trouble. All right, so moving on. That's what we mean by blood-brain barrier meninges, so there they are. And then the cerebrospinal fluid is demonstrated in blue here. So here they all are. You see your skin, periosteum, which is the skin on the bone, and you feel that right here. There it is. Go through the skull, epidural space. Here in the brain, we call it potential space because it's packed tight. In the spinal cord, we have a little area where you can insert the needle in. So it's more of a visible space down here, potential space up here. And again, potential in this case says, oops, I'm bleeding here. So now I'm pushing. If I'm bleeding, I'm going to push down on my brain tissue and cause harm. The dura has an outer layer, which is periosteal, sticking right to that, and then meningeal, which is beneath. We always do this tough on the outside, smooth on the inside. So that's what we're seeing there. Subdural, again, in the brain is potential because it's packed tight, but we have room in the spinal cord. Arachnoid, so little spider webs. And there they are with their little villus coming out between fingers. So arachnoid villus are little spider fingers. Love that. And then a pia is right on the tissue itself. And then our brain goes in this order from outside to in. Cortex, out, medulla, in. So cortex is always the outside of something. Medulla is the inside. The other words they gave us, well, they didn't give us for cortex, but the cortex is gray matter, meaning it's cell bodies and short axons. And all what we call medulla is white matter, meaning they're all strings going down towards you, back, and across. And this piece called the falx cerebri is a dip between the two hemispheres. It separates them, except for this one spot down here where we have what we call a corpus callosum, or a bridge. That's how we protect this whole thing. And as I mentioned, you won't have a skull on your sheep brain, but you will see the dura, and then this will be represented by the fluid that's in there. You have a hard time seeing this arachnoid, but you should be able to get at the pia if you peel at it. Meningitis is when we have inflammation here. That can lead to encephalitis, which is inflammation in the brain. Both can be deadly. And that's, I know, to get into college, you need meningitis evidence that you're protected against that, because it spreads in tight quarters. We're not a dorm college, but in places where people live in close proximity, they tend to have that show up. So why do we have CSF? The brain floats, reduces our weight quite a bit, protects us, slosh about, and it stabilizes the movement of nutrients and waste, and it helps clean us. The ventricles, which are just fascinating looking, there's a model in the lab of the ventricles, like they did a cast and took the brain away, and the ventricles are there. It looks like an alien coming at you. So the two up here, the first and second, are the laterals. So we just call them right and left lateral. The third one sits in the middle, and it's got a piece going right through it. So remember I mentioned the thalamus before? There's two thalami. There's a little piece connecting them. They are inside this fluid chamber. So they are represented here and here. And there's the fluid chamber. And then we get into a fourth ventricle, which dips behind the brainstem and into a little bit of the cerebellum. So it's two laterals, 1/3, 1/4, and then it dips down into a tunnel, which is like a straw, we call the central canal. So whenever you see a spinal cord picture, the very center of the spinal cord, you'll see a little circle. That's this. So if you follow it up, it leads to the fourth ventricle, leads to the third, and then to the two laterals. And they're just crazy looking. When someone has brain injury or bleeding in the brain, they'll often notice on the MRI or CT that the lateral ventricles are displaced, because the pressure will push, and you'll see one shunted or over, and those are signs of severe internal bleeding and swelling. So how do we make it? Oh, I know you're curious. So up here in our choroid plexus of the ventricle, choroid means a net-like, and plexus means net-like, so it's this netty area, that we're having a little setup of, right in the middle of the third ventricle, of using, pulling from the blood to create this fluid. It then goes into the lateral ventricles, and then to the cerebral aqueduct down to the fourth, and then it goes all the way down and around. So you'll see the arrows go around and down, and I don't need you to know specifically where they go, but it's just kind of a neat, if you're interested in the circulation, that tells you the whole thing. But I never test on that, it's too deep. So here's where we're getting to the fun stuff. This will be repeated in lecture and lab, and you just know the bone names already, so you're just gonna look what was underneath a frontal bone. How about a frontal lobe? What's under the parietal bones? Parietal lobes. Temporal bones, temporal lobes. Occipital bone, occipital lobe. There's 4 of them already. So the biggest, I shouldn't say the biggest, the easiest way to tell them apart is looking for what we call the sulcus, or an area where they're gonna have a fold in them. This one is very clear. They look like little ear flaps. So this is temporal. The frontal is farther back than you'd think it would be. It's way back here. And this thing we call the central sulcus is sometimes tough to recognize, but you're going to look for 2 gyri that are solid, and those are going to be pre and post-central. So pre-central gyrus is that, post-central gyrus is this. And all the models, we color them, which is awful nice. So you see one color here and opposite color there. That tells you you're in your frontal or parietal. And occipital's in the back, and he's going to go all this area, and then cerebellum, brainstem, and diencephalon come in. So we have 2 hemispheres. We would call them, so this guy's looking at you. This would be his right hemisphere. This would be his left of the cerebrum. We also have hemispheres in our cerebellum when we get down there, but we're not going to get into much detail there. They're very similar in function. And the right and left cerebral hemispheres are also similar in function, but they have different, we'll call them personalities. And that's what we mean by this lateralization. Each hemisphere is good at something and not so good at other things. So they work together to form a whole brain. And most of what we know about brains is when something goes wrong. So we realize long ago, not as long as we should have, we used to think the brain was a radiator and we cooled the heart, which obviously made us warm, that's obvious, right? But when we realized that, oh geez, somebody gets hit in the head and they're not the same, maybe something more is going on up there than just being a radiator. So early brain surgeries, where they were, historically they would go in with, screws and nails and open things up and let the bad spirits out. But let's say we're finally into the 20th or late 19th century when we're trying to experiment on things. We would have people who would have epilepsy, which would be so debilitating that they'd say, go ahead, open my brain, I just can't live like this anymore from the seizures. So some of the early things that were tried were, what if we cut this corpus callosum between the two sides, so that if a seizure starts here, we can't spread it and it'll save that. And that does work, but what it does is make these two brain sides work differently and not talk anymore. So if you've ever seen in abnormal psych, they generally show old videos, you can pull them on YouTube of the right left brain tests, where you would show the right eye something, and the left hand, and the right hand, and the left eye would all have different opinions of what's going on around them. It's pretty fascinating. It just shows that we're not communicating. Other things they would do, this is more behavioral control before we got good pharmaceuticals. You probably heard of prefrontal lobotomy. We take a little thing and just wiggle around. All right, how's that? Are they still being a bad person? All right, try it again. Okay, we're good. Not the best treatment, but it works. It makes you docile. We take out the frontal lobe and so on. So we learn about the brain when things go wrong. So what I'm getting at with this lateralization idea is that when we work with people who lost part of their brain or have had a stroke that affects one hemisphere or the other, because often strokes will be limited to one hemisphere because of the way the blood circulation is, that's when we really see these personalities show up. So if someone has a right-sided stroke, their left brain is now in control, and that left brain has a different personality than the whole brain would have, and certainly than the right brain did. And by personality, I just mean what we've shown here, different things. You get really deep into this, but I'll just do the general, just to give you a sense of how we understand basics. There's always people who will break the exception. For 97% of us, we are left brain dominant, not because we're right-handed or because we have left brain personalities, but because of that. There's a little area right here called the language areas. And there's two language areas that we use to determine which side of the brain is really kind of in charge of language. One is known as language comprehension, named after Wernicke. And up here is a motor region named after Broca. So Wernicke's and Broca's become the determination of where they are on something, which you'd only know if you had a functional MRI or had brain damage. We'd say, okay, that seems to be your dominant brain side because this is where your language centers are, regardless of handedness. Handedness is usually about 70% right-handed and 30% left, but that has nothing to do with that. It's where is this? And again, on 97% of brains tested, that's where it shows up. People who have it over here, it changes everything I'm about to tell you. You never know. So this first, we see this little strip here, that's your corpus callosum. If we snip that, these two sides work differently, they don't talk anymore. You'll notice, let's take the eyes first. The eyes up here, like the brain does, are going to crisscross. And we don't really know why this happens. We think it's to diversify and give us more protection, but this right eye is going to send half of its information here to where it's green, and half of it to there where it's purple. So what I see in this field over here, purple, is going to show up in my right occipital lobe. Again, I don't need you to memorize any of this, it's just fascinating to me. The right visual field shows up over here on the left side. Go figure. But both eyes control both. So half of my visual field from each eyeball talks to one half of my brain. Well, when we see a left-sided or right-sided damage, it's going to screw up how we deal with this. The one that really matters in losing vision is going to be the right hemisphere. And the way I describe this, and they call it representational versus categorical. So the way I would say it is categorical is logical, language, left, I put all those L's together. It's figuring things out by step. And because of that, we're looking at minutia. We're saying, all right, this goes here, put things there in categories. Over here, representational, we're saying, wow, look at the big universe. Everybody's connected. Very different way of looking at the world. So we see everything in the right hemisphere, but in the left hemisphere, we tend to see this, my focus. So when you open the fridge, well, usually it's men or your children, no need. Just look. So you scan, like, oh, there it is. But we don't see the whole thing. We're trying to scan, we're missing things. So this side of the brain is I see it all. This side is my scanning side. So this really shows up when you have a right-sided occipital lobe damage. These two purple fields disappear. So my world now is this. And it's half of each eye, but any of you guys over here don't exist to me anymore. So I'm over here, like, how's it going? Oh, nice to meet you. And I'm, whoa, where'd you come from? That is what you'll see on that, we call it heminopsia or heminopia. That is the loss of vision, of visual field. And that's pretty fascinating to me. It's not so bad on this side because the brain still knows to scan. So really what we're saying is we can change the input, but if the brain doesn't know that it's damaged or doesn't understand what's going on, it will make an excuse. The brain loves to do that, and deny and say, nope, we're all set. This is all the world I ever had. This left arm never worked. This isn't even my arm. I'm not worried about it. So these type of folks with right side brain injury tend to be extremely frustrating to work with because they don't understand what situation they're in. So with the left hemisphere in control, they've got a good verbal memory, they can speak, they often confabulate or talk nonsense because they are working on a memory that isn't matching reality anymore because they've lost the side of their brain. They control their right hand. Well, what's that all about? Well, again, we don't know why, but we crisscross right here through the pyramids, we call it decussation, right there, just before the foramen magnum, and we send all signals from the right to the left and left to the right. Coming in the same way, we crisscross it. So controlling my right hand is a very specific spot here, but this we could say the strip controls my entire body on the right side, and the strip controls my entire body on the left. This side feels my entire body on the right, and this feels it on the left. So losing this, I mean having this in control, I can't feel it or see this because I can't see over here, so I just deny it exists. You'll see them sitting on it, you'll see them wheeling down the hall and it's dragging along the wall. They'll stumble, they'll knock things, if they're driving, God forbid, the left side of the car is all scraped up, they just don't have a reality over there. You know someone like this. Right-sided, so we call them left hemiplegia. They're dangerous because they don't understand where they are. You also see the language comprehension is here and the right visual field, as I mentioned. So these folks talk a great game, but they're in denial. Over here, this is our memory of how things fit together, shapes, art, music, all this kind of stuff over here, the feeling things. Left hand control, musical ability hangs out here, recognition of faces and spatial relationships is here. So this is a huge component of how we see ourselves fitting into the big picture, where this is kind of a selfish brain over here. And we need both to operate. We need to be able to think in both sides, but this is where the networking comes in. Regardless of which side you tend to lean on more, and the joke is men tend to pick on their left brain and women tend to pick on their right, but I feel that women tend to use, if we're going to be gender specific here, tend to use their whole brain better than a male does. Men tend to say, do you want me to fix it or do you want me to listen? And if I have to listen, I have to sit on my hands because I'm trying to fix it. But if you want me to fix it, I'm not going to listen because I know what's best. So that's, again, me picking on men, but we have a harder time, I believe, talking between the hemispheres. So that's a goal, is to make a whole brain. And that just explains right left stuff. So way beyond the scope of the course, but it's fun to look at, and there's a lot of myth around it too, a lot of things, are you right brain or left brain? It's really, how do we operate on a daily basis, and what side have we specialized to solve problems? And do you tend to, we build networks in our brains, we tend to focus on one or the other. Like, well, this is what I do when I'm faced with this situation. But let's open our brains up and say, maybe there's other ways to look at it. The other lobe that we didn't mention is a fifth lobe. If we peel open the space, which is called the lateral sulcus between occipital and temporal, we find another guy in there. Hey, what are you doing in there? He's called the insula, which means insulated. He's deep in there. And he's on top of the thalami. So here's our thalami. The third ventricle is in between them. There's this little stalk. And around it, we have this tissue, which is, again, gray matter around white on top of that. So that's the fifth lobe that we don't see. The other sulci that we named, we already said central, lateral. We've got a parietal occipital, which splits parietal and occipital. He's really hard to see, but you'll note him in lab. And then finally, transverse cerebral just says, we're done with cerebrum, now we're moving to cerebellum. This is a cool shot of everything. We've peeled away, and coloring it helps a lot, because you can say, okay, this is frontal, this is parietal, this is occipital, that's temporal. Oh, and there's insula in there. And these two metal prongs don't belong, but they're showing us, letting us peek at insula. Because this is a left brain, and this is why they usually show it this way, we can slap the Wernicke area and the Broca area here, but he got lifted. So he's over here, but he's being lifted at the moment. So if we go back to when I said, remember I gave you a list of words that go on incoming and outgoing. So this is how it's arranged. We said before that incoming information is going up, and it's always going to be dorsal or posterior. So information that comes in from the body will rise up the spinal cord and enter behind the central sulcus. So in other words, everything that's not red or pink here is for sensation. All this is sensation, all this is motor, and this front part is thinking about motor or other things. So this is our thinking, meaning high level activity that we said, the prefrontal part. This is our premotor area, which says I have a motor plan, I know what I want to do, and it's all pre-programmed into my brain. So I just run a program, and then it goes to this, which goes straight down. So this is which we call our primary motor area, which is the prefrontal gyrus, is where we send information down. So when we say sensory up, motor down, sensory back, motor front, that's what we mean by that. So the spinal cord, if I look at it sideways, anything in the back of the spinal cord is rising, anything in the front of the spinal cord is coming down, and they both turn into a common nerve, which we'll get at later. But that's how this is divided. So the central sulcus is key to understanding brain function. If you say everything going forward is about thinking and moving, everything behind is about feeling our world, that makes a lot more sense. Another neat thing is that we move from thought to program to action down, but when we come up, we go to here first and then we spread it. So it's like here to go out versus here to go down. Just feel those flows there as you look through it. That also tells us that if I put this, and we'll see this in a minute, we'll call the homunculus. All of our body parts are mapped on here. So I have a very specific part that controls my hand and my mouth. And then if something happens to these neurons, I have trouble moving my hands or my mouth. This part right here, which is in front of my mouth, is the motor planning area for every speech sound we'll ever make. When we are newborns, we can make any sound that any human could, but based on what we hear in our environment, which is processed here, we will limit ourselves to the language of our native family. But if you're a multilingual household, you're going to pick up a lot more languages at birth and you can speak them fluently. If we try to learn it later in life, we'll manage, but it's not going to be as true as a native speaker would be. So this is what we call the motor speech area. When this area gets hit with stroke, we cannot make sound come out that makes any sense. People will make sounds, people will say things that don't make any sense, and they'll look confused when it comes out of their mouth. They'll often give you an unreliable yes and no. So you ask them something that I always have to concentrate. They'll do things like, no, no, yes, yes. Trust the head movement, not what came out of the mouth. If it's, aphasia, this is called. And then every other movement, we think about it, we lay it out in a pattern, and these patterns are called motor patterns, pre-motor, and this is going to be our recorded history of programs we run to move our body. And they will line up directly with motor neurons that will go down and talk to the muscles. Coming up for senses, we're going to rise up, land in the thalamus. There's two of them, and we can't see them here, but they're inside right under that insula. The thalamus is going to decide, is this worth telling my brain about? And if so, what I mean by my brain is you. So let me rephrase that. Your brain knows everything that's going on. We assume even in a coma, even when you're sleeping, it's catching everything. Just you don't catch vision when your eyes are closed, but you're aware of your environment. And you know that if there's a creepy sound in the house, you're like, oh, wake up, fully alert, because your brain is listening. So we assume people in a coma are also listening and feeling and everything, but they're trapped where they can't send messages back out. So what I mean by you is that your consciousness, wherever that is, we used to try to find spots. Well, it's right there. No, we don't know that. It's somewhere in there. Is it collected in the universe, or is it in your brain? We don't know. Consciousness is up for debate, but we know when it's not there. I guess that's the best way to say it. So if consciousness is you're alerted to things that your thalamus says this is worth you knowing about, meaning that most of the information that comes up, you're never made aware. Don't worry about it. I don't need to know. But that which is brought to your attention is brought either to the sensor area, which says this part of my body just felt something, or back here to this area where I'm going to see, and this eyeball saw something. Over here where I'm going to hear and taste. And these are all areas that are going to be alert. The one that I didn't mention is smell, because that does not have to go to the thalamus. I have little dangling things here, which are cranial nerve 1, might as well know him now, called olfactory. He's, again, on the, like, if this were the sheep brain, he's about that big on a sheep. Where an us, he's like that. But the olfactory bulb dangles into the nostril and lifts sent in goes directly to your insular cortex without having to be processed in the thalamus, which explains why scent is such a powerful emotional smell. Well, obviously it's a smell. Our emotional memories come out of things that you smell, like grandma's cookies, or somebody you miss, or a perfume that reminds you of a past love, all that fun stuff. And then the other areas are just set up for their specific senses, but they all talk to each other. And that's the other piece. Whenever we break the body down into regions, we have to remember that's us telling it that we're separated. In reality, it's all chatting with itself all the time. And a lot of this is done in what we call the association cortex. You see here, here, here. And this is saying, I've got all these feelings coming in, but what do they mean? Meaning is what we ascribe to say, this is a good feeling or a bad feeling. This is something I want to look at or I don't want to look at. Art appreciation, if you will. This is a sound that I like or a sound that I'm afraid of. And then this speech area is all here. So it's part listening, part touch, so we write and draw and listen, and part sight. The Wernicke area says anything that's happening out there that I consider language is processed here, and then if I can reproduce it, I deal with it up there. So someone with a stroke here, they don't understand anything you're saying to them. They just look at you like you're talking a foreign language. They're confused, they're worried. They may speak back, but again, they can't make sense of it. When they say something they don't understand it, we call it a word salad. Words just come out. If they have what we call global aphasia, there's no getting to them that way. You might be able to get them to write. Assuming, which is unfortunate, most of us are right-handed, so if your left brain is affected, you probably can't write with your right hand anymore. So now we got multiple issues for this poor person. So the other thing that, well, let me show you this before we go there. Motor areas are in the red and go down. Sensory areas are in the blue and go back, and the association areas are collecting it all in between. And what we're doing with the information we collect is making decisions on whether we're going to act on it or not. When we do act, we send a motor signal down and make the things happen that we've already talked about in skeletal muscle. And the information coming in, most of it comes from the spinal cord, but the cranial nerves that you learn are bringing it directly in from the brain, from the face, I should say. So eyes, nose, ears, tongue, all that's coming in directly without having to go to the spinal cord, but they do hit the thalamus except for smell. So the next few slides, and I'll stop us here, but because each one kind of talks you through each area, but this is a kind of neat way to look at it. If we just take off the frontal lobe and say, okay, here's my primary motor cortex. Here's my premotor cortex where I've saved motor patterns, which we call praxis. And then here's my ideas, and here's my motor speech. So they show us a woman playing guitar and singing. So we can see it all affected here. The premotor cortex has all the hours, years of rehearsal. I've strummed the guitar, I can find my chords, I know my sounds, I'm working on this, and it's all set. So even if I haven't picked up a guitar in a while, if I used to know how to do it, it should come right back to me. This is what we call muscle memory. It's not really the muscle's memory, but it's your brain remembering which muscles to use. So think of it that way when you say muscle memory. The muscles don't know, they're just doing what they're told. The primary motor cortex actually sends the signals to the actual muscles we need, but if you thought muscle by muscle as you moved, you'd never get anything done. Because there's, as you know, there's so many complex muscles, particularly in the hand, we can't think about those. We just say, do this, and it works. It's here. The visual area is making sure that the eyes are moving where we need them, which helps us learn. You notice we do a lot of watching when we're learning, and we rehearse mentally. And there's also the idea that you can see a brain light up when you're thinking about doing activity the same as if you're actually doing it, which is fascinating. So they'll have an athlete or someone who has to rehearse something and they're mentally replaying what they're going to do. The brain doesn't know the difference. The brain's actually doing it up here. So when you study your exams or you're doing your muscles, naming, do it up here. Just all the time, build your. And then finally, the motor speech area, as we mentioned, is going to control the specific muscles in the mouth and throat to let the sounds come out. Because sound is just breath coming up, but changing the pitch and the sound is all done with little tiny muscles in there. So we'll do the rest of the examples next time, but that sets you up for what each little slide will be about each little rope. And we'll call it a day. Thank you.